We must remove these bits off, otherwise the
Good
conference centre will get upset at the end of the session.
This is a completely different presentation. This is something future. So far we've been speaking about metals and everything, but this is more of biology. So what I want to do is just give a general update on at what level we are standing as an orthopedic community in relation to stem cells, essentially in cartilage regeneration. So I just want to give an update. Again, my name is Raj, I'm from Telford. So the regenerative medicine is one of the branches of medicine which is growing quite exponentially medical rate at the moment, because there's a lot of enthusiasm about it. But in truth, we were the first group to actually look into in terms of the stem cell role, because the initial studies were towards chondrocyte regeneration and differentiation using MSCs. So in truth, we were the forerunner of regenerative medicine, but gradually we have lost our control over it. So it's a rapidly expanding field, but the reason why it's rapidly expanding is we are able to understand better how the genetics can be applied clinically, and we are also able to understand the tissue biochemical and biomechanical properties, and at the same time we are able to simulate or synthesize these things so that we can apply for tissue modification as well as modeling. Now there has been exponential increase. So if you actually put a figure in PubMed research, you can see that this is when you put in the stem cell and chondrocyte. The first 10 years, before 10 years, there was hardly like 6 or 2 publications in a year. But as you can see, over the years' time, the publication has gone significantly higher, right between last 10 years' time. So in 2002, we had only 49 publications. We are in the mid of 2015, there is already 233, and we'll be well ahead of 400 publications by the end of this year. So the literature review, what I did was, I just searched in the PubMed in various keywords: stem cells, chondrocyte differentiation, cartilage regeneration. There has been at least more than 1,300 publications. And after I applied filters as humans journal, because most of them are animal studies, I was able to identify certain 51 publications which are sort of relevant for this talk. I also had a brief look at the Harrison textbook in medicine, in regenerative medicine. You'll be surprised that over the last 3 publications, the number of pages in the Harrison textbook has gone quite high. The recent publication almost has a separate chapter dedicated to this. And again, there is one textbook which was written by one of the orthopedic surgeons, which is on stem cells and cartilage engineering. So I had the opportunity of getting that book as well. I'm thankful to my librarians for that, and various textbooks on clinical and applied genetics in relation to this talk. So introduction, a lot of people understand stem cells as a very different thing, but there's a different definition that all cells can multiply, or in other words, they can self-renew, but it's a potency. So the stem cell should have self-renewal and potency. And self-renewal is able to produce identical daughter cells, and potency is its ability to differentiate into the different specific lineage. The common lineage what we want is either chondrocytes or osteocytes, or even certain adipocytes. So basically, you know about all this ethical business. The reason why it starts is, in the stem cell, how they are raising is, you have a fertilized ova, which forms a morula, then you have blastocyst stage. And that's the inner cell mass, and that cells, we can take it out, and then they can be applied for, they can be allowed to differentiate in various different manners, so that we can apply for clinical. So because we are killing an embryo, or us to harvest that blastocyst, or us to get that inner cell mass, there is always an ethical question that you're actually killing a potential living human being. So that's where the fight is about. So there are various stem cells. We get usually confused when this is stem cell, it's all same. They're different. The ultimate stem cell is a fertilized zygote, or that's called totipotency or ultimate potency. Then as it goes along, it becomes pluripotent or the embryonic stem cell. Multipotent is, it has various levels but not complete. Example is hemopoietic. Oligopotent is, we have one lineage, which is example is a neural stem cell. Embryonic stem cell is always a bit attractive because it has less HLA expression. Now this is the model with which all the stem cells ethicists or the tissue engineering principle is based on. It's called Waddington's epigenetic landscape model. Essentially what it means is a cell which is starts as a totipotent cell can, as it passes along various lineage and becomes differentiate, it loses its potency. So finally, it starts a totipotent cell in terms of either a B cell or a macrophage or fibroblast or muscle. So the purpose of these things is we are trying to reverse it. So, or in other words, you are trying to induce a pluripotent, because if you have to harvest a stem cell from a blastocyst or something like that, then there's an ethical issue. But if you're able to reverse a cell by nuclear reprogramming, whereby you are taking your nucleus out and putting a new nucleus, whereby either you can convert a differentiated cell back into a totipotent cell or a pluripotent cell, or directly into a lineage, for example, converting a myocyte into an osteocyte. So that's what the cell biologists are trying to do. So the normal development will be that lineage. But then reprogramming to pluripotency is called dedifferentiation, whereas if you're able to convert into direct separate cell, it's called transdifferentiation. So that's what they are all at the moment. So that's what most of the studies the cell biologists are enthusiastic about at the moment. Now that's one part. Now the second part is of the research is going on is where we want to get the stem cells from. The sources can be allogenic or autogenic. The allogenic attracts more favor because if you're able to harvest stem cells, then you can produce mass. So you can take it from a shelf and give it to anybody, like a O negative blood group. But for allogenic, you need a HLA typing, like a blood group typing. So that's one test. But again, it's a lot of fight about it. The other is autogenic. In autogenic, you are taking from the same patient. So it can be bone marrow, commonly followed by synovial fat pad. You can have cord blood, peripheral blood, spleen, kidneys, even dental mucosa, adipose tissues. There's a difference between adipose tissue as well as synovial fat pad. Harvest is different. But these are all the places where a lot of research has been performed. So the bone marrow stem cells, they are rich in both hemopoietic and mesenchymal. There's a difference between a hemopoietic stem cell and a mesenchymal. A mesenchymal, we can try to convert into chondrocyte or osteogenic lineage, whereas a hemopoietic, you are looking in terms of the hemopoietic for the hematologist purpose. So, but the yield is not great. So what it means is, if you take 30 mils of bone marrow, you can harvest about 100,000 or 100,000 cells. So you have to culture them. So all bone marrow stem cells, you have to not only collect them, but also culture them in order to get a decent number of cells. Obviously, the donor cell morbidity, and it's a painful procedure. Whereas in a synovial fat pad, these are quite expandable. There has a lot of research been intensive over this, because one, it is very easy to harvest, and it's higher yield. Even a 21 mLs will yield about 5.5 million cells. And there's a very lesser pain because it's we can do like a key, either from keyhole arthroscopically from the back of the patellar tendon, or if you are looking in terms of in a fat pad from either the trochanteric region or the abdomen, and a lot of people are happy to lose some fat. So there we can take from that. And the adipose tissues, in vitro studies have shown that they have better chondrogenic and osteogenic differentiation potential compared to the bone marrow cells. And they have stable pluripotency. One of the problem is when you're taking the stem cells and then you wanted to cultivate them, so you're allowing the cells to multiply. But after you're multiplying for few generations, they lose the potency or their ability to differentiate. Whereas bone marrow cells, they can only pass a few lineages or few passages. The synovial fat pad stem cells or adipose ASCs, they have stable pluripotency even after 10 passages in vitro. Now, not for you to call us, for if you are going to be let's say you are going to be a knee surgeon who is willing to do stem cell, you want to do a stem cell implantation for cartilage defects in young patients. You need to identify. You cannot really go and take some fat cells and then grind it and take some cells and then put it back. They have to have you have to have a cell biologist on your team who has to do certain marking, and they have to get it labeled, because these stem cells are now categorized under drug, as per a standard medication. It has to be certified as a medical product. So the ISCT, or the International Society for Cellular Therapy, has come up with certain criteria. So these are the criteria for our own knowledge. One, all the cells which you are calling a stem cell should adhere to plastic material. So that's one test they do. Second, all of them should have cell surface markers. This is how they identify and label the cells, which are CD105, CD73, CD90. These are cell surface marking at a higher level of lineage before they become differentiated. These are the markers which are expressed in a cell surface before they become differentiation. But at the same time, they also should lack expression of CD45, 34, 14, 11B, 17 in alpha, 19, and most importantly, HLA-DR. The reason is, once the cell is getting differentiation, it will start to express the HLA molecules. And once it's starting to express HLA molecule, it loses its ability to differentiate quickly. And the other thing is, it can become immunogenic. So that's the reason, if you are calling a stem cell a stem cell, it should not have an HLA-DR marker on its surface. And finally, all the MSCs which are you're harvesting, irrespective of what your use is, should have an ability to either differentiate in osteocyte, adipocyte, or chondrocyte. So, if you are going to become a surgeon with specialists in this interest, you need to have a cell biologist on your background who has to certify and issue a certificate that they have done these studies and that these are authentic stem cells which you can use it on your patient. So you cannot, unless, this is how US and UK and Europe works. Rest of the world, it's a different story. So so we have gone through two things. One, we've known the cells and the sources. Now, even though you might harvest a stem cell, how are you going to make them go into a chondrogenic lineage? What are the factors that will make a stem cell convert itself into a cartilage and not a bone or different thing put it into an environment? So these are the factors. So what in vitro, what we are using is, you are harvesting stem cell and you are putting in a medium, in a 3D scaffold medium with a TGF, transformation growth factor beta. Now the cell goes through various stages of condensation, chondrocyte differentiation, and chondrocytic hypertrophic maturation. But in order for you to cultivate, you should not allow the chondrocyte to become hypertrophic, because the minute it goes for the hypertrophy, it stops dividing and it forms bones, or it promotes into ossification mode. So that's one of the biggest problem for us to stop it. So during that, in the medium, if the studies have shown that the glucose, insulin, and the TGF-beta are required. TGF-beta is now commercially available for us to use. Now, finally, the most important step in order for a stem cell to become a chondrocyte is it has to start expressing SOX9 factor, and it's a gene, and that can be identified during a process of during the process of cultivation. We can gladly identify using PCR and other genetic diagnostic tests how the SOX9, whether it's induced and expressed, because the downregulation means it won't go into the chondrocytic lineage. So further test is, the culture has to be done in the low oxygen tension, because the higher the oxygen tension, it goes into osteogenic mode, or it forms bone rather than cartilage. And at all costs, the chondrocyte has to prevent cell flattening by using because the actin filaments, there is always in a scaffold or any biological, there is in the extracellular matrix you have the actin filaments. When the actin filaments are starting to compress the cell assumes that there is an external stimuli for stress. So the cell will go along the bone formation mode, not the cartilage formation, because cartilage has a different external stimuli, whereas a bone has a different external stimuli. So by preventing the cell flattening, they noted that the SOX9 is upregulated. Now the medium, there are other enhancers like fibroblastic growth factor 2, PDGF, they are all improves the presence of chondrogenic lineage. Now finally, when you are looking at a culture medium on a cell, how do we identify whether it is a chondrocytic lineage or not? Is the expression and production of collagen 2 and aggrecan, because none of the other cells will produce that. So how you are doing is, you are staining this medium for collagen 2 or aggrecan, and that's how the diagnostic tests have come up. Okay. So this is for not only for promoting the chondrogenic lineage, but also for identifying whether your cell is going in the right direction as you would imagine. So for example, when you are being a surgeon, in few years time, in future or something, this will, when your cell biologist is going to give you a report saying that I have provided this with this cell culture, and it is one of the tests you notice that it is producing collagen type 1, then you know that it is not going in the chondrogenic lineage. So you can gladly turn around and say that, sorry, we can't use it for this purpose because it's not going to produce cartilage, it's going to produce bone. So there is a lot of hurdles over the time, and it's being slowly being overcome. Rome was not built in a day. So this is the future. So it's going to take long time before it comes to a commercial retail level. So at the moment we have gone through we have some sort of agreement of suitable universally accepted harvesting culturing stem cells. In fact, there are various kits available for standard stem cell harvesting at the moment in both in US and Korea. The homing, how are you going to deliver the stem cells into the place where you want is another great debate. Whether we're going to do how are we going to deliver it? Is it an infusion or is it a local delivery by keyhole surgeries? That's one. Then the signaling pathways is also another hot topic. How we want to do, what are the pathways, interleukins, how we can influence them. But most of all, it's the factors which are promoting and de-promoting chondrogenic differentiation is what we are all enthusiastic about, especially drugs. Because what we identify is if a cell is being stimulated or down-directed by a certain stimuli, like I'm going to show you like a norepinephrine thing, then we are wondering whether we can give some beta blockers or something to suppress that in order to promote into chondrogenic lineage. So, and then again, with the synthesis of growth factors which are all commercially available, these are all gradually being overcome, and even the cost implications was quite high 10 years ago, is starting to come down. Still high compared to the standard procedures like miraculous fractures we are doing, but it's getting there. Now the scaffold is another big area of research at the moment. The scaffold is bad because if there's no point in having a stem cell without a scaffold, because you take some stem cell and you put it in a cartilage defect and you put a periosteal patch in the front, all that can happen is the stimuli and everything will be gone. In a few days' time, it will still produce fibrocartilage with the collagen type 1, which is of inferior capacity. So what we are doing is, we are trying to find out an injectable scaffold, or in terms of some sort of gel where you can put it in a defect and infuse some sort of some stem cells into that defect along with the gel. So what happens is the gel forms a solid and covers a defect, and gradually over time that will get incorporated with the new cell formation. So what we are looking in terms of either an injectable or implantable or even impregnated with the with cells. Some of the scaffold are quite bioactive in terms of you can gladly take some stem cells harvested and then mix it, and it forms like a small gel, and you can put it in a defect, and that's what they are trying to do. And transplantable, or in other words, we can make a synthetic cartilage extrinsically and then just transplant it internally. How it is possible is because of the nanofiber technology and electrospun technology, which are initially being applied for like a smartphone, the chip processors and everything, or even micro weaving of the clothes. We have taken those technology and they are able to create a 3D matrix whereby we are able to reproduce a collagen fiber orientation like a cartilage, like a 3D with a desired fiber orientation, and so that we can, with the predictable biochemical properties. So we are moving closer, but as I said, it's a long way there. So the matrix engineering is a completely different topic at the moment, apart from stem cell. These are very, very gaining attraction. So these are polymers, most commonly chitosan, it's called. I'm not sure whether anybody's heard it. It's chitosan. It's essentially from the chitin. What you do is from the chitin, it's a shell of the crustacean. You deacetylate. So it forms a polysaccharide, but it's still a bit of like a gel. It's not a gel, it's like a solid material. But when you're putting it at a certain pH and certain solution, you can make it soluble. So then you make that solution and you make it like you can form like injectable solution, and either by arthroscopy or something, we are assuming that we can fill the defect, and then in the room at the body temperature, that gel becomes solid. So the body temperature, so that's why it's called thermoregulated. So the chitosan becomes from a gel becomes solid in relation to physical, chemical, or biological stimuli. But the thermoregulation is what made into clinical trials at the moment in US. And apart from this, we are also looking in terms of myocardial, in myocardial infarct where the wall is not moving properly. They are trying to inject this and see whether that makes the wall a bit thicker in an infarct area, and neural regeneration. These all the other areas. Little bits just to add on. So there we have found that the stem cells not only has the regeneration but also has a paracrine and a trophic effect. How it does is it forms a neovascularization, promotes neovascularization. At the same time, it also the adipose stem cells, not the mesenchymal stem cells. The adipose stem cells has got an immunosuppressive property. So we are thinking there is a thought of utilization in autoimmune diseases. The other findings recently were the dexamethasone. They are at a certain therapeutic level. They are able to allow the MSCs into either a chondrogenic or osteogenic lineage. Certain weird case reports for meniscal repair when they are non-operatively, where they have given injections. But these are all from Korea, so we don't know how much of it's really true. Finally, this is one latest finding. It was found that the norepinephrine inhibits the MSCs towards chondrocytes, and what they do is they make them hypertrophy and forms bone, which is something we can predict because norepinephrine is more of a post-traumatic response. So when there's a bone breakage, the body wants to form bone. So the question is, if you're using it, should we use any beta blockers or something in order to allow, for example, when a patient is having a surgery, when you're having stem cell infusion, what we want to do is knock off that post-stress effect. So that's one thing at the moment is being explored. Obviously, more and more we do research, more and more we can get confused. So initially we thought that the adipose stem cells were the most important, produce more cartilage in vitro study. But the recent multi-center study, which was done all over Europe, showed that only the bone marrow derived MSCs had the ability to form both cartilage and bone in vivo. So the in vitro study showed something, in vivo study showed something. So it's a completely contradictory, but that was a good study. In fact, it was found that also. So limitations, I mean, over the time period, we would have thought of many things. These are the things which have been overcome recently. Surface markers has been overcome, suitable medium has been found, cell expansion, which was thought to be slow and difficult, now it's a 3-week, we can have a good culture quickly, scaffold problems gradually been overcome, limited proliferative passage, again, we have overcome that, aging of the cells, we have done that. Finally, there's how to form a normal article cartilage, how to get the ACs to produce not produce fiber cartilage is one reason. Now the future, in years to come by, will be the cellular component being integrated, incorporated into a scaffold to produce a biological function. The cellular components can be derived from various places like autologous cells, ES, IPS, or who knows, even allogenic standard cells off shelf. Scaffolds, so there are you can have either biodegradable synthetic, like your dissolving screws, polyglycolic acid, or in natural collagen. Minerals, further addition of growth factors can be done. Weakness, there is a lot of weakness at the moment. Most of them are animal based. Their publications are mostly from China, Korea, and Japan. Limited research. I mean, most of the studies are very much by the researchers. Not many of them are made into clinical trials. Contradictory evidences. Now, finally, although research can be very, very interesting, how much can we convert into a clinical application is a million-dollar question. I have to say that at the moment it is still at a very, very infant stage for us. And most of the publications, in fact, most of my references are not from orthopedic journals. They are all in various journals, Journal of Nanofiber Technology, Journal of Blood. So you look at it, you'll find, and surprisingly, Rheumatology, Oxford Journal of Rheumatology, they all have quite good points for us to understand. So I went to the library looking for the books. So all these books are for the standard orthopedics we know. That smallest book over there was the book by Khan, Mr. Khan, on I think we can make it out saying that tissue engineering and in orthopedic surgery. So this is where we stand at this moment. And my library is quite small, so you understand, you get the big picture. So the references, I have only put a few references. I didn't want to put the entire picture. Oh, yeah, I think that's it. Yeah.
I'm going to suggest, that was a super overview of where we are. I'm going to suggest that it's lunchtime now.
Yeah.
So if anyone's got any questions, they can ask over lunch. And if there is time later on this afternoon, I'll just say a few words about the exam as well.
Thank you.